I want to comment first on the durability question, because it's something that we really kept in mind when we designed this approach. And it's something that I think I want to be sure to sort of emphasize with using an AAV vector. And so we are using an AAV vector, but the payload is different than a microdystrophin approach. So in our AAV, it's encoding ARCUS nucleases that go in and correct the gene in the DNA, in the cell, in the patient's own body. That means that we're not dependent on persistence of the AAV long-term for continued effect. The edit happens quite fast, and then if the AAV were to be silenced or diluted out over time, the edit persists. It's a permanent edit at the DNA level. And I think contrast that with a microdystrophin approach where the therapeutic effect is being delivered in the context of an AAV vector. And so if that vector is silenced or lost over time, the durability of the effect may be limited. And so we really designed this approach with durability in mind, with permanence, long-term efficacy in mind. In terms of what that first in human trial could look like, yeah, that's a major goal of ours here at the MDA conference, is working with clinicians in the space who have a lot of experience treating DMD patients to understand what a successful trial could look like. I think we are trying to learn from those who've gone before us in terms of what are the right endpoints, what are the right inclusion criteria. It's too early, I think, to really say exactly what our trial will look like, but we are actively working hand in hand with the physicians in the space to really smartly design this trial.
Cassie, one last question for you. Just on a more general note, how much potential does gene editing approaches have in muscular dystrophies like DMD and Becker, and what value do they bring that gene therapies do not?
Yeah, I think we touched on this a little bit, just thinking about what the AAV is doing in the two contexts of a gene editing approach versus a gene therapy approach. And beyond just the thought of the persistence of the AAV, I think another key component when we designed this program was thinking about how can we ensure benefit for patients. Obviously durability, but how do we ensure that the protein that we make is going to be functional? And the reason that we landed on the 45-55 deletion strategy, it was really based on what's known in nature, known that this protein exists in humans, know that that protein retains the vast majority of the protein functional domains. It also retains the majority of all of the dystrophin interacting protein binding domains, which we know is essential for proper muscle function and for dystrophin to do what it's supposed to do within the muscle. I think that's pretty starkly contrasted with the very truncated versions of microdystrophins that have been designed in order to allow for delivery within the AAV vector. And so I think that a gene editing approach in the context of DMD has some major differentiators that could be really valuable for patients. We hope are really valuable for patients. Reasons to believe, in terms of the functional output of the protein and the durability of the effect. And then I think the ability to affect skeletal muscle at the satellite stem cell level is really just a huge potential long-term benefit for patients. And even if microdystrophins are able to target satellite cells, because they are not editing at the root cause of the DNA, their effect is dependent on the persistence of the vector. As those satellite cells divide, that vector expression, vector presence will get lost. And so I think that really to provide a long-term durable effect for patients living with DMD, satellite cell editing is absolutely essential. And so I think that really can only be derived from a gene editing approach. So I'm very hopeful that gene editing can be really transformative for this patient population, and I'm excited to continue to work to bring this to the community.